Excretion. What is excretion? For the proper functioning of the body, many chemical reactions occur in the cell, which is known as metabolism, and the end product of which is carbon dioxide, water, and nitrogenous waste that has to be eliminated from the body, otherwise will be harmful to the body. The mechanism involved in this process is known as excretion. The major excretory products are carbon dioxide, excess water, and nitrogenous compounds like ammonia, urea, and uric acid. Water and salts in excess of the body's needs are also excreted. Most of the acquired water is from food and drink, and some by metabolism, for example, the water produced during cellular respiration. Other excretory products include chemicals from medicines, toxic substances, and circulating hormones that have already served their purpose. Through excretion, organisms control osmotic pressure, the balance between inorganic ions, sodium and chloride ions, and water. It maintains acid-base balance. This process is known as osmoregulation. The process thus promotes homeostasis, a self-regulating process by which organisms maintain stability of internal environment. Carbon dioxide and water are produced in the process of tissue respiration. Nitrogenous compounds are formed from the breakdown of proteins and amino acids. Organs of excretion. Owing to the wide variety of diversity in animal kingdom, various organs are evolved for the process of excretion. In certain single-cell organisms like protozoa and porifera, excretion takes place through cellular membrane via osmosis or diffusion, and in others like amoeba and paramecium through vacuole. While in the case of higher vertebrates, the major organs of excretion include integument, gills, liver, and kidney. The major organs of excretion are listed below. Contractile vacuoles. Contractile vacuoles are the excretory organs of the coelentrates and the protozoates. Contractile vacuoles are spherical shaped organelles in which water enters. The vacuole then fuses with the membrane of protozoan and the water is expelled to the environment. Energy in the form of ATP is required for both the entry of water into the vacuole and for the emptying process. Protonephridia. True excretory organs. Discrete organs which have an excretory function are found in all animals above the level of coelentrates. The simplest of these excretory organs are found in the platyhelminthes and are called protonephridia. Protonephridia are excretory structures which exist as closed or blind-ended tubules and which do not connect with the coelomic cavity. The cell which forms the tip of the blind-ended tube is ciliated. If it contains a single cilium, it is called a solenocyte, whereas if it contains several cilia, it is called a flame cell. The beating of cilia transports fluid to the tip of tubules and exits the body via pores. As it passes through these pores, only small molecules pass and large molecules are retained back. And here, the process of ultrafiltration is first seen. Metanephridia. Metanephridia, sometimes called nephridia, are the organs of excretion found in many annelid worms. Fluid enters via the nephridiostome and urine leaves via the nephridiopore. Blood is filtered across the membranes of capillaries and the fluid produced enters the coelomic space. This is another example of ultrafiltration, since only water and molecules of small molecular weight enter the coelomic fluid, whilst large molecules such as proteins remain in the vascular system. The initial urine which is formed passes along the nephridia, where its composition is altered by the process of reabsorption and secretion. The result is the production of a urine which is hypoosmotic, that is, less concentrated to the body fluids from which it was formed. Malpighian tubules. The excretory organ found in insects are called Malpighian tubules. It is a closed tubular system located in the hemocoel of insects. The number of these tubules varies from few hundreds to many thousands. The open circulatory system of insects leads to no pressure-driven filtration. Rather, they utilize active transport of potassium ion and chloride ion for the formation of urine. The urine thus formed then enter the midgut, where further reabsorption of important molecules like protein and water takes place before it is finally ejected out into the external environment through a pore. The nitrogenous waste excreted by insects are in the form of uric acid, which is very less toxic, hence requires very low amount of water to be eliminated from the body. Crustacean green glands. Crustaceans, for example, crabs and lobsters, have excretory organs called green or antennal glands located in the head region. The green gland consists of a blind ending sac called the end sac connected to a tubule, the nephridial canal, which terminates in a region called the bladder. The bladder exits to the external environment via an excretory pore, which is situated near to the base of the antenna. The end sac is surrounded by coelomic fluid, which is filtered to produce the initial urine, which lies within the gland. As with all excretory structures, the composition of the urine at this stage is similar to that of the body fluid hemolymph from which it was formed, with the exception that it contains no substances of high molecular weight such as proteins. As this fluid passes along the nephridial canal, water and other solutes are reabsorbed. Unlike the bladder in mammals, for example, the bladder associated with antennal gland is also capable of reabsorbing substances. Vertebrate kidney. The vertebrate excretory organ is a pair of kidneys located in the abdominal cavity. It is a bean-shaped organ made up of thousands of tubular structures called nephrons. Nephrons are called the functional unit of kidney as the ultrafiltration, reabsorption of important macromolecules, and urine formation occur there. Types of excretory products. In living organisms, protein are used for many cellular mechanisms, and the metabolic end product of protein is nitrogen, which cannot be eliminated in its free forms. Hence, it is converted into ammonia, urea, or uric acid before it is eliminated out of the body. Ammonotelic. Ammonia is the chief breakdown product of amino acids, which mostly takes place in the liver and kidney via processes such as amination and amidation. Ammonia is a very toxic substance which requires immense amount of water to be excreted from the body. Hence, it is the excretory product of animals that live in water like fishes, some reptiles, and few mammals. And thus, these animals that excrete ammonia are called ammonotelic organisms. Ureotelic. Urea is derived from amino acids of protein metabolism. First amino acid is converted to ammonia and then it is converted into urea via ornithine cycle, also called as urea cycle. Urea is highly soluble in water and is much less toxic than ammonia. Hence, the organisms that excrete urea is called ureotelic. This process occurs in the liver where the enzymes secreted by it converts ammonia into urea. The steps in ornithine cycle are as follows. Carbon dioxide and ammonia are first converted into carbamoyl phosphate. Carbamoyl phosphate is then converted to citrulline. Aspartate is added to citrulline in the presence of magnesium and ATP and is converted into argininosuccinate. Fumarate splits from argininosuccinate to form arginine, while fumarate enters Krebs cycle where it forms malate and oxaloacetate. Arginine in the ornithine cycle releases urea to regenerate ornithine and cycle continues. Enzymes involved. Carbamoyl phosphate synthetase 1 converts ammonium and bicarbonate into carbamoyl phosphate. This is the rate limiting step in the urea cycle. This reaction requires 2 ATP and occurs in the mitochondria. Ornithine transcarbamylase combines ornithine and carbamoyl phosphate to form citrulline. It occurs in the mitochondria. Argininosuccinate synthetase condenses citrulline with aspartate to form argininosuccinate. This reaction occurs in the cytosol and requires 1 ATP. Argininosuccinate lyase splits argininosuccinate into arginine and fumarate. This occurs in the cytosol. Arginase cleaves arginine into one molecule of urea and ornithine in the cytosol. The ornithine is then transported back into the mitochondria for entry back into the cycle. Uricotelic. The third major means by which nitrogenous waste products are excreted is in the form of uric acid. Animals which excrete uric acid are said to be uricotelic. Animals which are uricotelic include insects, birds, and reptiles. The advantage of excreting uric acid is that very little water is lost with it. The urine of birds, for example, is a semi-solid white sludge. It may be considered to be an adaptive mechanism to living in the terrestrial environments where the conservation of water may be at a premium. The metabolic pathway by which uric acid is formed is very complex. Essentially, ammonia is converted into a substance called glutamine, which in turn provides the structure around which uric acid may be synthesized.